Maybe just coming through.
Okay.
But I'm good to keep up with the conference, the lectures and everything. Is there anything else I need to be doing?
If you got questions, you shoot me an email. Happy to go over stuff.
Perfect.
Okay.
Thank you.
Yep. Okay, welcome everybody. I know that my audio is working for those tuning in at home. I don't know what they did to the thing. I think they unplugged it and plugged it back in, which when in doubt, do that, I guess. All right, maybe something else magical happened, but I didn't see that. So we left off last time about halfway through this chapter. My intentions are to cover chapter 8 today, and then we're going to take Friday off. I have an appointment that I thought was happening later in the day, but it turned out it is earlier. So, and I will post a message, but no class Friday. There shouldn't have been class today. That's what I thought as I was getting messages from my kids' schools, and then from ISU. I just knew ISU, they're not going to close. The only time we close is really when students are on holiday in the first place, and we're like, oh, well, now we'll do a snow day, right? Which, whatever, right? Okay. Questions or concerns before I jump into this stuff? So I left off, I know we talked about this first bit. This was the tail end of glycolysis, pulling in a sugar molecule, phosphorylating it. So it takes a little bit of energy to make energy. We phosphorylate that sugar molecule, we break it in half, and in turn, with a little bit of manipulation, we get 2 3-carbon molecules of pyruvate. We pop those phosphates off of those pyruvates, or the phosphoenol pyruvate, PEP, and that is what we describe as substrate level phosphorylation. This is happening where our substrate, the PEP, is going to donate that phosphate. There's a kinase, and I believe oh man, I gotta mute. Okay. So substrate level phosphorylation takes a phosphate, puts it back onto an ADP to regenerate or recharge it into ATP molecules. And this depends on the type of organisms, but in some circumstances, when they think about being able to metabolize, this might be as far as they go. And that's really not so much energy that they're able to acquire in this process of manipulating these glucose molecules. There are many other opportunities to truly try to harness as much energy as we possibly can. And that's really what we want to think about as we move through and talk about respiration, as we go through aerobic and technically anaerobic respiration as well. We can see similar amounts or pretty close to the same amounts of energy that can be achieved with anaerobic respiration. Really, in those circumstances, we're just using some other substance besides oxygen as the final electron acceptor. When we think aerobic respiration, oxygen is the one that accepts that, and we generate water molecules as a result. So we'll go through and think about those steps as we progress through, all right? So it also depends on what kind of organism we're talking about. We're in microbiology, we're focusing on bacteria, but there are similar things that are occurring when we're talking aerobic respiration, whether it be a bacterium or our own cells, right? The locations of these reactions are going to be a little bit different, but similar sort of an idea, right? Glycolysis happens in the cytoplasm of all cells. Doesn't matter, bacteria or a eukaryote. After we've done glycolysis, then it does matter who we're talking about. And in a prokaryote, all of these reactions are really happening in the cytoplasm in the cell, or when we think electron transport mechanisms on the membrane, plasma membrane of that bacterium, that's where those proteins are going to be linked up, and we will push hydrogen ions into that inner membrane space between the membrane and the cell wall of the bacterium, right? If we're talking eukaryotes, mitochondria are where these processes are happening, right? And inside of the mitochondria, which were once aerobic bacteria, these reactions are occurring on the inside of the mitochondria in the matrix, and then electron transport happens on the inner membrane of the mitochondria, pushing stuff, the hydrogens, into that inner membrane space between the outer and inner membrane, like the bacteria is doing in between its membrane and cell wall. Same sort of an idea, establishing hydrogen ion concentration gradients that are supercharged, if you will. We can make energy out of that. All right. So before we get to that bit, we want to think about dealing with those pyruvates and really shuttling them into these reactions. So we're moving right into citric acid cycle or the Krebs cycle, and we want to further manipulate these pyruvate molecules. We're not thinking glucoses anymore. We've already broke them in half, right? In order to do that, the first thing that happens is an association with an enzyme, coenzyme A, that is going to snag that pyruvate, but it gets manipulated really. It gets one carbon dioxide broken off of it, and now we've got a 2-carbon molecule that's going to be shuttled into the citric acid cycle, all right? That's the idea. And again, depends on who we're talking about as far as where this is happening. But in our bacteria, will be in the cytoplasm where citric acid cycle is occurring. Matrix of the mitochondria is where it would be occurring in a eukaryote. So this enzyme, coenzyme A, and when it's linked up, as we see here, that's how it truly looks in a chemical model. Our 2-carbon molecule gets attached to this coenzyme A. We call it acetyl-CoA when it's linked up, and it brings it in to the citric acid cycle. There, for simplicity, we show one round of this stuff, but we've got 2 pyruvates that were generated, so we're going to do this 2 times to deal with both sides of that, both pyruvates, right? Both acetyl groups get pulled in, right? So as this happens, and there are other steps that we're not showing in this initial figure, multiple enzymatic steps that help to transfer these molecules around. We'll see some details in 1 second with this, but they basically drop off this 2-carbon molecule to a 4-carbon molecule. It's called oxaloacetate. Oxaloacetate, when it snags these 2 extra carbons, becomes citrate, a 6-carbon molecule, and the cycle is named after it. All right, that's what we are building. All right, when I talk about this stuff, doesn't really matter micro or biology stuff. When I talk about this, and I looking through more of this, we've got a little photosynthesis in here as well. Both of these cycles, they're not the same, but it's a similar sort of an idea. The citric or the Krebs cycle, and in photosynthesis, it's the Calvin cycle. They're similar to like a cooking situation where we've got sourdough starter that you make your dough. You take a little pinch of that dough after you've done all your stuff, and you throw it back into the starter, and your volume in that starter pretty much stays the same, all right? The next time you go to make some sourdough, whatever, you pull the starter culture out, you take a pinch of it out, throw it into your mixture. That's your yeast and bacteria that you're adding. And the dough rises and you make your stuff, all right? Citric acid and the Calvin cycle are kind of like that. It's constantly going. It's constantly got this little starter culture, if you will. And as long as we've got an input of molecules, the cycle just keeps going, all right? We're generating energy. But really, what we're generating are electron carriers. That's more what we're really trying to get here, because these guys are going to participate in the last step, the electron transport chain. And this will give us our oxidative phosphorylation, not substrate level, all right? So although we get some ATP or GTP, depending upon what kind of cell we're talking about as we're generating. This is a guanosine instead of an adenine. Similar sort of an idea, all right? For most purposes, it's ATP that we truly think about. We get a little bit, all right? But we get a lot more of our electron carriers, right? So these are some oxidation reduction reactions that we're really looking at, or redox reactions, coupled reactions, right? And so with our little positive here, NAD plus, when it is like this, it's in the oxidized state. And when it snags a hydrogen and an electron with it, but we don't show that. When it snags this bit of energy, if you will, we say it's reduced, right? And with these redox reactions, this is a pretty good point right here. Maybe if all of this stuff works, I never like using this pen, but we'll see. So it's a redox reaction. And I think of this, it's oxidation and reduction, right? And it's coupled. Oh, there's a button there. That's an L. Writing. Okay. So one thing is oxidized, and it in turn can be reduced, and it just moves these things around, all right? And I remember some of this as oil rig. Oxidation is loss, that's oil, and reduction is gain, all right? Oil rig helps me remember what's happening, all right? If something is getting oxidized, it's losing something, all right? Losing electrons, losing its cargo, all right? The hydrogens are what we're going to lose as we oxidize that molecule, getting it back to its oxidized state, the NAD plus, right? The same with the FAD. We don't give it a positive, but FAD, it snags two hydrogens, all right? I think of these like, you know, little molecular taxi cabs or Ubers, whatever you want to call them, all right? And you need a ride somewhere. They pick you up and then they dump you off. Once they dump you off, they're ready for business. Back seat's open again, right? I could pick up more cargo, right? Once you're in there, right, then that car, it's reduced. It's got its load of passengers, all right? And our NAD, we could think of it like that. It can take one passenger, whereas the FAD, a little bit bigger, it can haul two, all right? If you've got a taxi cab that's only big enough to haul one person or two people, you better get a different car, right? That's what I think about, all right? But these things are constantly getting oxidized and reduced, shuttling hydrogens and the energy within this, these electrons, shuttling these from the citric acid cycle and bumping them over into the electron transport chain that we're going to talk about in a second, all right? So as we do this, we're bringing in a 2-carbon molecule. Our acetyl group is a 2-carbon molecule. It's got oxygens on it. It's got hydrogens on it. And the hydrogens, we've just snagged, right? We pull all of those hydrogens off. And the carbon and the oxygens, they go off in this process right here. This is where we lose all of our carbon, although, I mean, we've technically lost 2 CO2s as we were dealing with the acetyl group to bring it in to the citric acid cycle. There was 2 CO2s that we lost for each of those pyruvates, right? 1 CO2 per pyruvate. 2 of them brought in, we lose 2 of them, but we're doing this twice for both of those pyruvates. So there'd be 4 carbon dioxides that we've given off. At the end of the citric acid cycle, all of the glucose, all of the carbons in that molecule have been dealt with, and it's been exhaled as CO2 or given off as carbon dioxide bubbles if we're talking some other thing besides us, right? This is where it's all lost, right? And like I said, a little bit of ATP, but the bulk of it truly are these electron carriers, right? Are NADs and FADs. And it's nicotinamide adenine dinucleotide and flavin adenine dinucleotide are the 2 compounds that serve as these shuttles, right? Similarly, when we talk photosynthesis, it's the same players, but there's a phosphate in those molecules that participate in shuttling hydrogens that we don't see in other organisms. So this is the same idea that we were just looking at, or we're just talking about. But it's got a little bit more detail, right? And I did say last time at the end of the OpenStax text in the appendices, I believe it was Appendix C, it shows all of these different types of reactions with all of the enzymes that are governing these mechanisms. If deep down biochemistry is your passion, I recommend you take a peek at that, right? Maybe it is, you don't know, right? I don't know. Somebody in here could truly, deep down, that's what they really want, right? You will learn lots of this, the details of this in biochemistry courses, because really all sorts of enzymes governing each of these steps. And that's a lot of what we think about in biochemistry in the first place, is how enzymes do it. And can we calculate rates of these reactions? How quickly or slowly do they happen, right? So I really like this figure. I think it, I mean, I don't like seeing all of the steps in here, and I don't, that's not something that I find myself geeking out over, all right? But what I like with it is all of the red boxes, because it shows us that when we go through this process, we don't simply just take in a sugar molecule and break it down and get energy and electron carriers. We're going to break it down and potentially utilize some of these substrates, some of these products from one step to the next might get shuttled off into some other anabolic reactions. So everything in red would be a building or an anabolic reaction. Whereas as we're moving through this other stuff, we're catalyzing, right? We're breaking things down. But in red, building, all right? So just depends on where we're at and what sorts of stuff we are needing within that cell, as far as do we pull some of these out, right? With that said, there are other potentials where we think about hopefully eating more things than sugars, right? And in turn, we could think of the opposite, where these things in red, as we're going out to build them, we could be putting them into this system as we start to metabolize and break some of these molecules that we have used to build to feed back in to this, right? Dependent upon the circumstances, right? So oftentimes, really when we're thinking these reactions, we'd see 2 arrows, both going in both directions, reversible types of reactions, different enzymes, but the potential to build it or break it down, right? Which makes sense, right? So there are numerous steps in here, lots of amino acids that we could potentially start producing that in turn serve as building blocks for proteins. We can generate some nucleotides. fatty acids and sterols, thinking membrane components, more amino acids and nucleotides, and then some other groups if we're thinking other molecules, porphyrins and chlorophylls, and heme groups when we start thinking about transporting molecules like oxygen within our blood or in the chloroplasts, these chlorophyll pigments are the players in harnessing the energy from the sun for photosynthesis in the first place, right? So dependent on, again, like I said, the needs, we start generating this stuff, right? And you can see the cargo getting loaded up in a handful of our electron carriers. And these two bits right here, as we are churning through from the oxaloacetate to the citrate, this is where we start oxidizing some of those molecules, reducing our NADHs, generating one carbon dioxide, and we do the next one with the alpha ketoglutarate as it's moving into succinyl, and that in turn pulls off the next carbon dioxide as we reduce this other NAD. So these are the points in the citric acid cycle where those last carbon dioxide molecules are released, right, as a byproduct. And then it's energy production. And then basically like the sourdough starter stuff, we're recharging, right? So we're coming back to this, manipulating this molecule around from succinate, that's a 4-carbon molecule, up to the oxaloacetate, still 4 carbon, but rearranged, shifting the structure of this molecule. And basically at this point, it's ready to snag another 2-carbon molecule. And the cycle keeps going, all right? That's the Krebs or the citric acid cycle. So the next bit is really thinking about all of that cargo, all of those hydrogens that we were able to pull off of there, all right? So a single glucose molecule, C6H12O6, there's 12 hydrogens that we pull off of there, all right? The CO2s get exhaled, all right? It's the hydrogens that we've pulled off of these molecules, and they were covalently bonded to those carbons, and in turn, lots of energy to build those covalent bonds, and that energy could get harnessed. So the hydrogen ion and a little bit of supercharged up electron get snagged in this process of Krebs on that NADs or FADs, right? And they shuttle stuff in here to this electron transport chain, a complex of proteins, kind of assembly line like. They are studded very close to one another on the membrane. And it, you know, like I said before, plasma membrane of that bacteria or the inner membrane of mitochondria are where we see these proteins, all right? And the idea is for those molecules that were reduced, the NADs and the FADs that have become NADHs and FADH2s, as they come in contact with these proteins, this is basically like the Uber getting you to your destination, and you get out and you're done, right? It drops the cargo off, right? And the red, as it's moving through here, are really the charge, the electrons that are moving through these complexes. And we're using this energy to drop the payload off and to shove it against its concentration gradient into the, in this case, the, you know, inner space between the plasma membrane and the cell wall when we're talking bacteria, right? So outside of the cytoplasmic membrane, this is inside, all right? So we start pumping a bunch of hydrogens in here against their gradient. And as we shuttle these, the FADs give more energy. That gets the first bit of energy re-excited. We keep these things kind of going. And as we look at this, it's kind of charging as we move through, as we're seeing it going up. And then finally at the tail end, that last bit of energy, not the hydrogens, but the energy, the electrons that were being transported through these protons, This little jolt of energy is enough in an aerobic organism to take oxygen molecules, O2, that we have inhaled in, and to split it apart into two oxygen atoms, and we dump hydrogens right onto it. It becomes the final acceptor. So one O2 molecule, when we blow it apart, gives us two molecules of water, two H2Os, right? We oftentimes use it as a half oxygen, right? Half atmospheric oxygen to show we get one water, right? But you're technically getting two out of this mix, right? As we go through here, and hydrogens link up, right? So we push a boatload of hydrogens into this space in between wherever we're talking, all right? And now you've got a big concentration gradient. These hydrogen ions charged up, and in turn, they're able to do some work. So this purple pear-shaped guy in the membrane is an enzyme called ATP synthase, and we see it in all sorts of organisms that can go through respiration, all right? We also see ATP synthase in photosynthetic organisms, in membranes generating ATP in the chloroplasts, right? And we also see it in their mitochondria, all right? So the hydrogens, as we get a concentration gradient, this is really kind of like a faucet, all right? Turning it on, turning it off. If we've got a good charge of energy, if we got a lot of hydrogens up in here, now we're able to do something. When we turn the faucet on, this protein can twist, right? From an on to an off as it is manipulated based off of concentration gradients. As it turns on, it allows these hydrogen ions that were forced in here against their gradient to move down their concentration gradient. This is just simple diffusion, all right? At this point, from high to low concentration, the hydrogens flow down through this protein structure, and it takes that energy as they're moving through this protein, and it recharges the ADP molecules to make ATP. This is oxidative phosphorylation. We oxidized in the Krebs cycle, we oxidized those acetyl groups, pulling the hydrogens off. They lost stuff, right? Hydrogens. We reduced our NADHs after we oxidized the acetyl groups. And as they come into these mechanisms, they get oxidized as they donate their hydrogens to in turn flip back around and get reduced again, all right? That's the idea, all right? So as we percolate through here, we've got a boatload of energy that's available, and we charge up a good amount of ATPs if we can do oxidative phosphorylation, all right? And this is aerobic respiration. We've got our oxygen down here, all right? So cytochrome C oxidase is the one that is the last one that splits this water molecule as the energy moves through there. If we're talking an anaerobic system where we are still respiring, it's basically the same sort of an idea, except this last little bit isn't a cytochrome. It's not something that's an oxidase, all right, that's acting on oxygen. It's acting on something else, right? Maybe a sulfur, all right, and generating hydrogen sulfide as a response with that. One sulfur putting two hydrogens onto it. Similarly, like we see with this oxygen, right? Just depends on what you're truly able to breathe, all right? There are all sorts of crazy things bacteria can do, all right? And we'll talk a little bit about this later, but I'll give you just a hint right now, all right? Some bacteria are aerobic, and they flat out have to be, all right? And we would call them obligate, all right? Obligate aerobes. If you don't have oxygen, you can't grow, right? But lots of them might be able to tolerate different things, all right? And there can be some facultative organisms or microaerophilic organisms. The microaerophiles can tolerate oxygen, but they don't like flat out full oxygen. They want to be kind of masked the littlest bit. So little reduced oxygen, and then they're happy, right? The facultative ones, they're a little different, right? And a facultative organism, we oftentimes say facultative anaerobes, right? They can go either way. And if you have oxygen, they will go through and respire. But if they come into a circumstance where oxygen is depleted or missing, well, they'll ferment. They'll go through different pathways, right? They still survive, right? But it's a different mechanism of working with it, all right? So handfuls of ways, all right? Our own cells can do some similar stuff with fermentation. If muscle cells get starved of oxygen, they can potentially ferment and make, in our case, lactic acid as a byproduct. We'll see that in a second, all right? But when we're talking respiration, aerobic, it's oxygen, and anaerobic, it's some other substance down here, all right? That is used as the final electron acceptor. But similarly, the amounts of energy generated, pretty close, right? And this is where we get the bulk payoff of energy that we can achieve, all right? So this, as we're moving, and this, I believe, right here, through the electron transport chain is where we're getting the 24 ATPs. This bit right here is really Krebs, all right? Although we're showing this as we're breaking it down with the carbon flow. The electron transport chain is where we get the bulk payoff, right? 24, all right? Glycolysis and the net stuff, all right? We get a little bit of energy, but as we get to the tail end of all of it, we've got a potential for 38, theoretical, all right? Usually it's less than that. With this theoretical bit, sometimes a little bit of that energy is needed, utilized to get our coenzyme A shoved back out so that it can keep working, all right? There's some other things that it does, all right? And also theoretical, because we could flip it back to this, and as we're moving through here, there are many things that we're shuttling off, so that's going to impact the amount of ATP that we're truly maximizing from every single glucose we're pulling in. It's not 100% fully oxidized every time, because some of it gets shuttled down other pathways. That's what gives us some of this variation, all right? So when it comes to fermentation pathways, it kind of depends on the circumstances, depends on who we're talking about, all right? I mentioned lactic acid. That's something that happens in us. It happens in other eukaryotes. But some, like yeast cells and some bacteria, can go through and generate other molecules like ethanol. So alcohol is a byproduct instead of lactic acid, right? Some bacteria generate lactic acid like our friends that make yogurt, right? Lots of those are lactic acid bacteria, and through fermentation pathways, they take in the lactose sugars, metabolizing those and generating some lactic acid that gives yogurt its kind of tart taste as a result of that. With alcohol fermentation, instead of using the pyruvate as the final acceptor to generate lactic acid, we're going to take it and metabolize it just a little bit more. So we get one carbon dioxide molecule popped off of our pyruvate and a 2-carbon acetaldehyde molecule as this byproduct. The acetaldehyde is used as the final electron acceptor, our NADH, donating hydrogens to it, all right? We just pull them off. Now we're putting them back, all right? And in doing that, it manipulates this 2-carbon molecule, dumping the hydrogens onto it to create a ethanol molecule, 2-carbon molecule, all right? With the lactic acid, it's a 3-carbon molecule, and the NADH puts it right back onto it, and it manipulates it into lactic acid instead of ethanol, right? Just depends on who we're talking about, all right? I have heard some stories, whether or not this is 100% true. I feel like it probably is. There are some people that I guess I would call mutants. And instead of going through and, their muscles starved of oxygen and they generate lactic acid, they go through and do this process and they generate ethanol, right? And so with some strenuous activity, they end up finding themselves intoxicated because they robbed their cells of some oxygen, and we're still going to perform. And in the process of doing so, you also got intoxicated. Crazy, all right? That truly, I think, is a problem, right? You got a drinking problem, right? But you weren't drinking, right? That is crazy, all right? I'm sure there's some details about that. I bet you there'll be a pack back. I can feel one coming on. This bit it's not something you're going to see in lab, not like this, but you're going to see some of it, all right? The API test strip is basically a boatload of different biochemical tests, different types of sugars or other molecules, but lots of sugars that we can inoculate with one bacterium and then basically see, can it use this or not? And can it potentially do other stuff, like maybe produce gas or not? A couple of these things may help us to differentiate types of bacteria, right? This is stuff that we kind of think of like classical microbiology, using biochemical tests, which we're going to show you a good chunk of them in the lab, using biochemical tests to truly kind of pinpoint types of microbes, right? There are other techniques that we think about with like maybe some genetics, extracting DNA, sequencing stuff, and getting specific answers. Oh, this is E. coli or whoever it might be, all right? But in classical micro, it's more phenotypic types of responses. Can they feed on this or not? And based off of that, it can clue us in, all right? So this API system, you would inoculate each of these wells. There are others that are kind of like little tubes that have an inoculating needle that you pull out as you've got the bacteria on it, and it gets every one of those wells inoculated pretty much simultaneously, all right? You cook it up for a day or two, and then you pull it out and look at it. And they come with a book that shows you positive and negative reactions. And then you start coding stuff up, all right? So as we look at this, we've got 3 wells and a triangle, another 3 wells and a triangle. As we move through here and look at these in their specific little groupings, we start giving them pluses or minuses or 1s or 0s, depends on the specific systems, all right? And then you basically add up the in between the triangles, right? So you're going to come up with what do we have here? 1, 2, 3, 4, 5, 6, 7, a 7-digit code. Once you have looked at all of these wells and you've done a little bit of math, right? You get a 7-digit code that you look up in this big, thick book that has all sorts of organisms. And based off of the coding, it tells you this one is E. coli, or this one is Photobacterium damselae, all right? Whatever it might be, all right? All sorts of different potentials, all right? This stuff is pretty cool. And I think it's interesting to kind of see some of it happen. We had different ones than the API. that we used, and there were issues. Oftentimes not saying really what it should have been. And part of that has to do with the bacteria we work with in the lab, all right? Most of the organisms that we are working with are what we call attenuated strains. They're weakened. If they are a disease-causing organism, when you attenuate it, you basically cultivate it over and over and over. And in the process of manipulating it over multiple generations, if you will, it starts to lose some of its characteristics. Things like the ability to make you sick. We'd like to get rid of some of that stuff so we could work with it in the teaching labs and show these organisms, but have a kind of level of knowledge that these things are safe to work with and not something that's going to cause severe illness, right? Deep down, I feel like if you ingest some E. coli, you're probably going to get the craps, right? It's probably going to give you a bit of diarrhea, all right? But it's not something that you're going to be sitting there like, oh, I'm going to die, right? It's not something that's super dangerous, all right? So by attenuating some of these organisms and they lose some of their properties, it impacts some of these biochemical tests. And what should be a definitive positive to clue us into something might wind up being a negative, all right? So it lends to some problems, all right? And we typically don't mess around with these things because it usually doesn't work quite like we like it to, right? The biochemical tests that we use in the lab, those things seem to typically give us the results that we really want as we are going through there, all right? So you'll be doing some of those biochemical tests soon, or are you doing them right now? Do you know what's happening in lab? They're coming up, all right? Soon. I will tell you when you do your oxidase tests, they are little, they look like little windows, and you rub a little bit of your bacteria onto one of the squares of those windows. When you do that test, you want to make sure that you've got a good glob of bacteria on your loop to put it on there, because if you just barely have the littlest bit, it may not be enough to really give you the positive reaction that you should be getting, all right? And you also need to look at it pretty quickly. They say within 30 seconds, all right? If you rub stuff on there and 5 minutes later, you go back and look at your oxidase test and it looks purple, well, you can't really say that that's really accurate if you didn't see it change to purple in 30 seconds, right? Because it can oxidize just with the air and give you a false positive, right? Not enough could give you a false negative, right? Too long of an incubation time, just sitting on the bench, right, could give you a false positive. Either way, not so good, all right? So shifting over briefly, we're going to look at photosynthesis, but I don't want to get super bogged down with this stuff, all right? We will talk the littlest bit, all right? I feel like this is more truly biology stuff, all right? But our little cyanobacteria or their precursors truly set the stage for this in eukaryotic cells in the first place. So we ought to think about it the littlest bit, all right? Here we are looking at a chloroplast, right? And we've got all of these alkaloid membranes. This is where the chlorophyll pigments are studded on these membranes, kind of like the inner membrane of a mitochondria, sort of the same idea. This is where light reactions are happening, and the synthesis reactions, the production of sugar, happens in the fluid that these membranes are basically floating around in, all right? The fluid I like to think of as sugar water, because that's where we're making the sugars, all right? So in this reaction, we use energy to excite those molecules. That energy can break molecules apart, like water molecules. We can also potentially manipulate other things because it doesn't necessarily have to be oxygens that are coming out of this, all right? photosynthesis has a handful of different potentials. Before we get to truly thinking about the oxygen revolution and this being the byproduct oxygen gas, right? That's why it's an A, because there can be some variations with it, all right? But with what we're thinking about, water molecules pulled in, the energy splits those molecules, we get oxygen gas as a byproduct, and those hydrogens that were on the waters, we're going to use to reduce our NADPs into NADPHs, right? That's the idea. Like the reverse of what we just talked about with cellular respiration. We generate a little bit of ATP, and both the ATPs and our cargo, the hydrogen ions, move out of the thialkaloid membranes into the stroma, where they interact with carbon dioxides that are getting pulled in, and we start pumping out sugars or other molecules, right? Just like how we showed in the Krebs cycle, other things being generated besides our energy. It's the same idea as we look at this. There's other stuff that can potentially happen. So in a eukaryote, we've got the chloroplast with the thialkaloids. In our prokaryotes, it's a precursor, infoldings of the plasma membrane that basically have these pigment molecules studded on them that do a similar idea, like looking at in here, just differently, all right? And this, again, a prokaryote, a little bacteria that's photosynthesizing. They don't have these structures because the bacteria itself ultimately became something like this when it was engulfed in some ancestral eukaryote millennia ago, all right? So this, a bit of photosynthesis as the light energy gets in and starts hitting those chlorophyll pigments, the light harvesting centers as they're picking up this energy, getting super excited. They transfer energy to this reaction center and it excite an electron, bumping it up to an outer orbital where it's supercharged. It wants to do something, all right? And it in turn moves it, shuttles it into electron transport chain, charging up or reducing our NADPHs. That's the idea. Like the opposite again of what we were seeing with respiration, all right? So oxygenic and anoxygenic, again, something else, not oxygen, or excuse me, not water, as our acceptor down here. And here we see oxygen given off with oxygenic as we're splitting water. But over here, it could be something different, right? Not that. And we generate other substances, hydrogen sulfide, hydrogen gas, or other molecules. And potentially, as it's showing here, water, but we're not splitting the water, we're adding it to those oxygens. Different, right? So oxygenic or anoxygenic. And the early photosynthesizers started off like this, right? And then transitioned to splitting of water molecules and generation of oxygen. And those early photosynthetic prokaryotes were those that initiated really this oxygen revolution. And we start seeing aerobic bacteria that have the capacity to deal with this toxic oxygen molecule that's being generated. And here we are today, right? So multiple photosystems moving electrons and shuttling things, really like the reverse of what we thought about with our respiration. So lastly, there are handfuls of cycles as we move through here, and lots of stuff that I'm going to just quickly gloss over. Carbon, as we think about it, the atmosphere is a big reservoir for carbon dioxide, but there are other bits as we think about fossil fuels with carbon tied up within there. And, you know, we can see the burning of some of these increasing CO2 into the atmosphere. Some of it with regular respiration and photosynthesis, we see some balance, but we also see some imbalance with the excessive production of CO2 through the burning of fossil fuels. Not a balanced equation, right? So nitrogen, I mentioned before, in the atmosphere is N2 gas, and with bacteria that fix it, we can manipulate it into ammonia products that plants can tap into. And then with the denitrification, releasing nitrogen gas back to the atmosphere, another cycle, and microbes participating in fixing and denitrifying those substances. Sulfur, another where we think about atmospheric sulfur and the potential for organisms to utilize anaerobic organisms to utilize some of these in their mechanisms. And we're not going to get bogged down with all of these cycles. This is the last bit. And when I first was looking at this morning, as I was flipping through, getting lecture loaded up, I was like, why do I have Neisseria at the very end of this lecture? And then I remembered at the start of the chapter, the little case study with the person that's got issues, right? It's meningitis, and it all comes back full circle. Now I remember. So Neisseria meningitidis, gram-negative diplococci, when we think about meningitis, this is the bad one. Not that others aren't bad either, but it's definitely a serious bacterial infection, all right? And there are, as we see here, some regions in Africa that are kind of ripe with this bacteria. There are all sorts, as I'm looking at this, this is epidemic risk and endemic in yellow. A lot of this we would really kind of describe as the fly belt, where there's all sorts of infectious disease, mosquitoes, huge vectors for all sorts of things, not our Neisseria. But when I first looked at it, I was like, oh, we're looking at malaria. No, we're not. We're looking at meningitis, right? And that, again, like I was saying, was the focus of the case study in this chapter, was talking about an individual that had traveled, I believe, to Africa and then contracted this upon returning. All right, that's the end of that. Remember, no class Friday. I will post on Moodle that. And I also will post both of these lectures yesterday or last time and today, because I realized I didn't post Monday's stuff. All right, farewell. Have a great weekend. We also don't have class Monday. Holiday. It's now you're like, he did that on purpose, but I didn't. All right.